Hi, thanks for coming along to my talk today, where I will be presenting some of our work in characterizing thalamocortical connectivity. So, the thalamus, shown here in orange, is a subcortical structure with widespread connectivity to the cortex. This widespread connectivity facilitates both neural communication from the peripheral nervous system to the cortex, as well as communication between cortical regions, giving the thalamus a key role in shaping whole brain dynamics. As such, by studying the organization of the thalamus, we may gain insight to how these dynamics are shaped. Investigations of thalamic organization often focus on its nuclei. These approximately 30 areas are associated with distinct cytoarchitectural and connectivity features. However, different nuclei can have the same patterns of cortical connectivity. This is most easily observed when looking at how parts of the thalamus are clustered by structural or functional connectivity. You can see here that these clusters do not align with the boundaries of the nuclei. Several principles have been proposed to try and explain how patterns of cortical projections can vary within and across nuclei, and how this relates to underlying molecular architecture. One such study looking at these principles was conducted in the mouse by Phillips et al. In this study, they clustered thalamic nuclei by their similarity in gene expression. They found these clusters were arranged along a medial-lateral positioning. Furthermore, they performed a PCA of the gene expression of each nuclei and found the first principal component varies systematically along this axis. This plot shows principal component score against each nuclei's medial-lateral position. What this shows is that regions with a similar medial-lateral position had a similar pattern of expression for genes associated with this component, while nuclei at the most extreme medial and lateral ends had more distinctive patterns. Moreover, position along this axis, or gradient, was associated with differences in cortical projections, axonal morphology, and laminar targets, as well as differences in electrophysiology, suggesting this axis represents an underlying principle of thalamic organization. Given this, we wanted to see if these results obtained in a mouse model can be extended to a human model as well, particularly in regards to characterizing the cortical projections of any such thalamic gradient. To do this, we first defined a set of seed coordinates, these black dots, throughout the thalamic volume, shown in orange. Given the gradient in the mouse study was defined based on transcriptomic information, we endeavored to do the same. We used data from the Allen Human Brain Atlas, which measured gene expression across the brain, including the thalamus. What we did is, for each seed, we can measure the expression of a particular gene at the voxel where that seed is located. By repeating this process for approximately 2,000 genes previously identified as being highly expressed in the human brain, we can build a matrix of seed by gene expression values. Additionally, as we wanted to examine cortical projections from any resulting gradient, from each seed we conducted probabilistic tractography to 250 cortical regions of the left hemisphere. We can then calculate the number of connections to each cortical region. By repeating this for all seeds, we can make a matrix of seed by cortical connectivity values, and we repeated this across 76 participants of the Human Connectome Project to get a group average connectivity matrix. When this matrix is appropriately normalized, we can then combine it with the gene expression matrix we defined earlier. This matrix therefore represents each seed's connectivity across cortical regions and also expression across genes. We can then use this to define gradients informed by both feature types. To extract these gradients, we can use everyone's favorite dimensionality reduction technique, principal component analysis. We use principal component analysis because it yields us two matrices, a matrix of principal component scores and one of principal component loadings. The scores indicate axes of variation in both gene expression and cortical connectivity across thalamic seeds, while the loadings reflect patterns of cortical connections and gene expression for each component. We can use the data from these matrices to answer the following questions. Firstly, does the dominant axis of variation map onto a medial-lateral thalamic gradient, like that in the mouse? And secondly, if this gradient exists, what pattern of cortical projections emerge from it, and is this pattern associated with cortical function? So, to begin, we first examine the dominant axis of variation. The dominant axis, the one which explains the most variance in the original data. We can see here that the first principal component, or PC1, explains approximately 30% of the variance in the original data. If we extract the associated scores for this axis and project them onto the corresponding seed position and surrounding voxels, we see a gradient emerge. To understand what this shows, regions in red, which are located laterally, have similar patterns of connectivity and gene expression to each other along this particular axis. Similarly, regions in blue, which are located medially, have similar connectivity and gene expression patterns to each other, but these blue regions are the most dissimilar to red regions, and everything else resides somewhere in between these two extremes. So what we observe from this is a gradient which runs medial-laterally across the thalamus. We can further confirm this by correlating each seed's PC1 score with its medial-lateral position, and again, this shows that this component varies medial-laterally. So we have this medial-lateral thalamic gradient. However, we do not know if it corresponds to the same one that was observed in the mouse. We can test if the genes which are associated with the lateral end of our axes, which are those with the largest loadings, are enriched for homologs associated with the lateral position in the mouse data, and we can do the same for those with the lowest loadings, which are associated with medial regions. We find that across the 100 genes most associated with lateral regions in the human data, that they are significantly enriched for mouse homolog genes. However, those genes associated with the medial end of this axis were enriched, but not to a significant level. Taken overall, these results do suggest that the homologs of genes associated with the medial-lateral gradient in the mouse are associated with the medial-lateral gradient in humans. Finally, we wanted to see if the projections from this thalamic gradient corresponded to a distinct cortical patterning. We can look at this by firstly ordering seeds by their principal component scores along this first component and plotting their cortical connections as defined by tractography. We can see as we move from the seeds with the highest score to the lowest that the pattern of connectivity varies from posterior sensory areas towards anterior frontal ones, suggesting cortical projections from the medial-lateral thalamic gradient roughly map to an anterior-posterior cortical one. Furthermore, if we take our principal component loadings for the cortical regions and we project those onto the cortex, we see this same cortical gradient emerge. This shows that the medial-lateral thalamic gradient maps to a distinct cortical patterning. Additionally, as the mouse results show that medial-lateral gradient corresponds to distinct electrophysiological profiles, we wanted to see if the cortical projections of thalamic regions also corresponded to distinct functional patterns. Previous work has identified that connectivity between the thalamus and cortex relates to intrinsic timescales. Intrinsic timescales indicate whether a particular brain region's intrinsic fluctuations in neuronal signals tend to be fast, as indicated in red, or slow, as indicated in blue. We can see that visually the pattern of intrinsic timescales appears to align to a cortically derived gradient. If we directly correlate each region's PC1 loading and intrinsic timescale, we see a strong correlation, showing that cortical projections of the medial-lateral thalamic gradient are associated with fundamental patterns of cortical function. So, in summary, we find a dominant medial-lateral thalamic gradient defined by transcriptomic and structural connectivity features. Genes strongly associated with this gradient appear to be homologs to those which define the medial-lateral thalamic gradient in the mouse. We also found that cortical projections from this thalamic gradient correspond to an anterior-posterior cortical gradient and fundamental patterns of functional activity. Additionally, these thalamic and cortical gradients also correspond to fundamental developmental axes. Neurogenesis progresses along an anterior-posterior cortical gradient, with anterior regions beginning neurogenesis before posterior ones. In the thalamus, development progresses along a lateral-medial gradient, with lateral areas beginning neurogenesis before medial ones. Of course, there are more aspects to neurodevelopment, but this does suggest that the properties of these gradients, including their connectivity patterns, may be induced by this pattern of neurogenesis. Curiously though, the thalamic regions that begin neurogenesis first are connected to cortical regions that start neurogenesis last, and vice versa. Exploring how this particular pattern of connectivity arises is therefore of interest, and given the increasing availability of developmental imaging data, this is something we are keen to explore next. Finally, to recap, the principal axis of transcriptomic and structural connectivity in the human thalamus occurs along a medial-lateral gradient, which is potentially conserved across species, is associated with distinct cortical patterning and function, and may also reflect sequential development.